Ladies and gentlemen, I greet you all and I want to welcome you once again to our class. In today's class, we are not going to be talking about organism. We want to advance into another layer of biology, which is called the molecular biology. And some of the things I'll be saying here may look a bit complex, but if you pay rapt attention, I strongly believe you will see understand what I'll be saying. So today we want to talk about molecular biology, molecular biology and genetics. Some students have been writing me to talk about this aspect of biology that seems to be a mystery. Sometimes because it involves uh, the study of biology at the molecular level, some students tend to find it very difficult. And I'll be taking this step by step, one after the other. And the first thing I want to look at today is the chemical nature of DNA. When we talk about DNA, we are talking about the nucleic acid. We have two different types of nucleic acid. Nucleic acid, we have the DNA, we have the RNA. So these are the two naturally occurring nucleic acids. Other examples that are synthetic abound, but these are the two naturally occurring nucleic acids. And when we talk about DNA, we're talking about deoxyribonucleic acid, and RNA is ribonucleic acid. Ribonucleic acid. So if you remove the DOZ from that of DNA, it will give you RNA. And that also explains the fact that RNA is a transcript of DNA. RNA comes from DNA. Or RNA is produced from DNA. There are some few uh, occasions where DNA can be synthesized from RNA, and that is what we call the reverse transcription. If we have time, we can also look at that when we'll be talking about the central dogma. So, the chemical nature of DNA. So, DNA is usually bounded with proteins, and when separated from these protein components, DNA is made up of a repeating unit that is called the nucleotide. I want to repeat what I said. I said that DNA within the cell is usually bounded with protein or by protein. And when you separate, during DNA extraction, some of these proteins are, are separated from DNA so that it can extract a clean or a pure DNA. We'll have several enzymes that are used in doing this, proteinase K and the rest of them. So when you separate DNA from these proteins that bound them, you will now discover that DNA is made up of repeating units that is called the nucleotide. So these nucleotides repeat themselves or are found in a repeating fashion. The nucleotides are found in a repeating fashion. So uh, there's so many things to talk about, but let me see how I can. We'll have what is called the nucleoside. The nucleoside. I will have what is called the nucleotide. The nucleotide. So I'll just give you a difference by the time I explain the components of this nucleotide. Remember I said that DNA is now made up of repeating units that is called the nucleotide. Look at this. Assume I have this as a nucleotide. This is another nucleotide. This is another nucleotide. This is another nucleotide. This is another nucleotide. I'll still give you the I'll still give you the molecular structure of this nucleotide. This is just for explanation. So DNA is made up of this repeating. You can see this is repeated, repeated. This is this is repeated. This is repeated. This is repeated. So we can number it 1, 2, 3, 4, 5, 6. And sometimes we'll have about 1,000 to 10,000 nucleotides, like the type you see in messenger RNA. Messenger RNA is made up of about 1,000 to 10,000 nucleotides. The one with the smallest number of nucleotides is the transfer RNA that has about 80 nucleotides. That has about 80 nucleotides. Okay, so I want to just a minute. Let me see the okay. So this nucleotide I've mentioned now, the nucleotide is made up of the nucleotide the nucleotide is made up of three different components. We have the first component of the nucleotide, which is called the phosphates. The phosphates. The second component of the nucleotide is called the sugar, and the third component of the nucleotide is called the nitrogenous bases. Nitrogenous bases. Please don't forget these three: the phosphate, the sugar, and the nitrogenous bases. So conventionally, the phosphate is written in this form. The phosphate is written in this form. And not forget that this is the same phosphate that is found in ATP, the energy currency of the cell, which is called adenosine triphosphate. So in that case now, we have three different phosphate molecules. Three different phosphate molecules. When one is depleted, that is when energy is released, then it reduces to ADP, which is adenosine triphosphate. So it's same phosphate. So this is the first one. Then the second one is the sugar. The second one is the sugar, the sugar molecule, the sugar molecule. And that reminds me, please note that the kind of sugar found in DNA is called the ribose sugar. Ribose sugar. And ribose sugar means that it has 5 carbon atoms. Ribose, it has 5 carbon atoms. That's the kind of sugar. The kind of sugar that has 5 carbon atoms is found in the DNA. And the structure is like this. So here you see H, you see OH, you see OH, you see H, you see H, you see OH, you see OH, you see H, then C2OH. So this is called the pentose sugar. Sorry, the kind of sugar found is pentose in DNA. I mentioned ribose. Sorry, it's called the pentose sugar, not ribose. I'll still tell you the difference between the pentose sugar and the ribose. So the kind of sugar that is found in the nucleic acid is pentose sugar. A pentose sugar is a sugar that has 5 carbon atoms. So the sugar can now be deoxyribose sugar or ribose sugar. So deoxyribose sugar is the one found in DNA, then ribose sugar is the one found in RNA. So, but this is a pentose sugar. And why is it a pentose sugar? Because it contains 5 carbon atoms. So let's number them. This is 1 carbon atom, 1. This is 2. This is 3. This is 4. This is 5. So you can see 5 carbon atoms. So this is the general structure for the sugar. But then, if we want to talk about the sugar found in DNA, because what we are looking at is DNA. Don't forget the chemical nature of DNA. So we want to look at We will say DNA is made up of a repeating unit that is called a nucleotide. And we now broke that nucleotide into three different groups that make it up. The first one is a phosphate. So we're looking at the sugar. The sugar. So the kind of sugar that is found in DNA is what they call deoxyribose sugar. Ribose sugar. Some people call it 2-deoxy. Two. Some people write it as 2-deoxy. And the reason is this. If you look at the sugar that is found in DNA, there's an elimination of hydrogen atom in such a way that what you now see is what you now see is there's an elimination of oxygen atom, not hydrogen, oxygen atom, in such a way that what you now see is H. Hmm? In such a way that what you now see is H. What you now see is H instead of so we now see, look at 2-deoxy. This is oxygen. 2-deoxy. That means that at the second carbon atom, this is the first carbon atom, this is the second carbon atom. At the second carbon atom, there's an elimination of oxygen atom at the second carbon atom. That's the meaning of 2-deoxy. So if we eliminate that oxygen atom, that means that this becomes the structure of the deoxyribose sugar, and that's the sugar that is found in DNA. But it's not the same in ribose sugar, because in ribose sugar the OH is still intact. The OH is still intact. So by the time we remove the OH, then of course it gives you the kind of sugar that you see in DNA. Just a minute, because there are so many things to talk about. I want to be constrained to what we have here. Okay, so let's continue. Then the base. Let's now discuss the base. Let's now discuss the base. The base. The base, like we said, the nitrogenous bases. We have two different types of the nitrogenous bases. We have the purines, which is made up of adenine and guanine. We have purine. I hope you can see what I'm writing. Okay, you can see it. We have purine. Purine is made up of adenine. Also have guanine, the guanine, adenine, the guanine. Then we have pyrimidines, pyrimidines. The pyrimidines are made up of the cytosine, the uracil, and the thymine. So these are the nitrogenous bases. The purines are called the adenine and guanine. The pyrimidine are called the cytosine and the uracil. Now, let's go back to, before I begin to draw the structure of the nucleotide, let's go back to the nucleosides. Difference between nucleoside and nucleotide. So, nucleoside plus phosphate is equals to nucleotide. So when you add the nucleotide and a phosphate, then you add the nucleotide and a phosphate, it gives you nucleoside. That means the nucleoside itself is made up of sugar plus remember we said we have three parts of a nucleotide. We have the phosphates, we'll have the sugar, we'll have the nitrogenous bases. So these three, this number 2 and number 3, that is sugar plus N, will give you sugar plus N will give you nucleoside. So by the time you now add plus P, then equals to N, which in this case is nucleotide. So that is just the basic difference between the nucleoside and the nucleotide. The nucleoside and the nucleotide. Nucleoside and nucleotide. Okay, I think the next thing we'll now look at is to draw the next we'll now look at is to draw the structure of a nucleotide. The structure of a nucleotide. The structure of a nucleotide. So like we said, we'll have a sugar, this is the base, so I'm still drawing the nucleotides. I'm still drawing the nucleotides. Just want to be sure that everything here is correct. And from the look of things, everything here is very correct. So this is the structure of a nucleotide. And we have three different regions which are called the 5' and the 3'. So one edge of the nucleotides is called the 5', then the other end is called the 3'. And we're going to make reference to these primers by the time we begin to talk about DNA replication. DNA replication. Okay, let's continue. If you look at this structure, then you see that we have two different nucleotides. We see a repetition of only two nucleotides. Don't forget I said that when DNA is separated from the protein component, it's made up of repeating units that is called a nucleotide. And in some nucleic acid, depending on the nucleic acid, you can have thousands of these nucleotides. So, but in this case, we only have two. And each nucleotide will contain those three components I mentioned. So let's look at this now. So the whole of this is one nucleotide. Then the whole of this again, the whole of this again is another nucleotide, the second nucleotide. So in this nucleotide, we can see those three components I've mentioned. This is a phosphate group, this is a phosphate. Then from this CH2 down to the whole of this is the sugar group, and this is the base. So the same thing applies to here. This is the phosphate group, then the whole of this, the whole of this is the sugar, then this is the base. And like I said, this base is made up of the adenine, guanine. Because of how complex the structure is, and because of space, I will not go into drawing the base. The alphabet B is enough to indicate what we are talking about, which is the base. And don't forget that depending on what the structure you're drawing, the base that is found in DNA is a bit different from the one that is found, I mean the basis. And the only difference is that in DNA you see the thymine, and in RNA you see the uracil. That is just a basic difference between their bases. So I want to now talk about the bonds that are found in the nucleotide. The bonds are found in the nucleotide. This is already 21 minutes. I want this thing to be too long. Want to talk about the bonds found in nucleotide. Now we have a bond called the glycosidic bond, glycosidic bond. So glycosidic bond is the bond that links the sugar and the phosphate. And don't forget that when the first time I drew the structure of the pentose sugar, you could see OH here. You could see OH here. You could see OH here. You could also see OH here. The question now is what happened to the OH? Those OH within these regions were used in the formation of these bonds. For this particular one, because it's DNA, the O was removed or is not found in the sugar of DNA. That one is understandable. But the O, what of the OH or the base? The reason is because the OH was used in the formation of this glycosidic bonding. If you're good at chemistry, you have a clearer understanding of what I mean. Then in the same vein, there's also OH here initially, but the OH is no longer there. That is because the OH was also used in the formation of bond. This kind of bond is called the ester bond. The bond between the phosphate and the sugar is called the ester bond. Then the bond between the sugar and the base is called the glycosidic bond. So with this, we have uh, explained the nucleotide. The nucleotide. So you see that nucleotides are linked together by the formation of bonds. They are linked together by the formation of bonds. You see formation of bond between the sugar at the three carbon atom, eh, three carbon atom and five carbon atom. That is how, assuming we have assuming we have what was it called again? Assuming we have another nucleotide, it would have explained this thing better. So nucleotides are linked together by the formation of bond between phosphates. At the three carbon atom. This is 1, 2, 3. 3 carbon atom, then 4, 5. 3 carbon atom and 5 carbon atom. Nucleotides are linked together by the formation of bond between phosphates at the 3 carbon atom and at the 5 carbon atom. So let's forget about the nucleotide now. With this, I believe So let's look at okay. I will now talk about the I will now talk about the backbone. Hmm? The backbone. Having looked at the structure of DNA, the structure of the nucleotide, and the different components that make up the nucleotide, we'll now look at the nature, how the backbone of DNA looks like. So we have this as a base. So we have this. This is the base. So we have this as the sugar. Let's use alphabet. I think alphabet will be better. This is S, 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 S. So look at this now. If you look at this, you discover that DNA, you can also see those three components. Those three components are mentioned in the DNA. At the center, you see the bases. Then at the strand, mm, at the strand, you will now see the the components that make up the strands of the DNA, or which is also what we call the backbone of the DNA. So the DNA backbone, or the backbone of the backbone of DNA is made up of the sugar and the phosphate. The sugar and the phosphate molecule. These are the two that make up the backbone of DNA. This is also similar if you have your DNA, you have this, you have this. So in this case, you see that the bases are found here. The bases are found here. Then you now have the backbone. So within these strands, you see the backbone within these strands. You see the backbone within these strands. You see the backbone, the DNA backbone. S P S P P P P SP. So within this, these are the backbone of the DNA. So the backbone of the DNA is made up of the sugar and the phosphate. So we can continue to write it indefinitely. Sugar phosphate within all this backbone. Whereas within the at the center, you see the bases. At the center, you see the bases of DNA at the center. Okay, I will talk about this. Let's delve more on this basis. Let's look at a typical DNA strand. Let's look at a typical DNA strand. So if we have this as a DNA strand having A and maybe T, G, C, T, G, C, then A. For instance, you could have this as a single strand of DNA. With this strand, you can predict what the other strand can be. It's very easy to predict what the other strand can be. It's easy to predict what the other strand can be. So if we have this as a strand, then you can predict this. So if we have that A, if we have A here, you can easily predict that what we have here is T. But before I go on with this, there's something I want to tell you, which will help you in this prediction. There's a man called Chagaf, and according to the man called Chagaf, that brought about the Chagaf rule, he said that purines must pair with pyrimidines at the ratio of 1:1. And this is similar in, all organisms. The pairing of purine must be in the ratio of 1:1. Remember, purine is Ag. Remember that pyrimidine is C, then U, and T. So what Chagaf is saying is that A must pair with T and G must pair with C. It's a standing rule, and this is how it occurs. So having this knowledge of this kind of pairing, then you now know that A must pair with T. Then if we have A here, this should be T. If we have T here, we should have A here, we should have C here, we should have G here, we should have A here, we should have C here, we should have G here, and we should have T here. Have you seen it now? So this is the pairing rule. And apart from that, you should also note that there is a double bond that exists between A and T. A double bond also exists between T and A. Then we have triple bond existing between G and C. That's the guanine and the cytosine. Then we'll have another triple bond here. So anywhere you see T and A, you have double bond. We'll have triple bond. We'll have triple bond. We'll have double bond. And this kind of bond is called a hydrogen bond. The hydrogen bond. The hydrogen bond. So it is the hydrogen bond that bonds, bounds or bonds, whichever way you want to put it, that bonds or bounds the basis, the complementary basis, the complementary basis. Okay. So like I said, I want us to be taking this slowly. Let's look at another area to look at. So let's look at DNA replication. DNA replication. And like I've mentioned, the two strands of DNA are said to be complementary. They complement each other. That is because if I know what is in one strand, I can know what is in the other strand. A minute, this is 33 minutes. So let's see how far we can go. DNA replication. So we want to look at how the DNA replicates. How the DNA produces exact copies of itself. And of course, this process of DNA replication occurs during a cell division, the meiosis and the mitosis. When this occurs, it is important that the DNA replicates. So let's look at some of the enzymes that are involved in this DNA replication, or by extension, how the DNA replicates. How the DNA replicates. All right. So the first thing I want you to know is that DNA replicates in a semi-conservative manner. However, we have different schools of thought about how DNA replicates. We have the conservative manner, the school of thought that projects the conservative manner. Then we also have the ones that project the semi-conservative manner. But I want to concentrate on the semi-conservative manner, which is most popular. And according to this method of DNA replication, DNA kind of produces replicates DNA replicates in such a way that the offspring strand contains one parental strand and one newly synthesized strand. The offspring strand contains, for every offspring strand, it will contain the one parental strand and one newly synthesized strand. So that is the doctrine or the conservative manner of DNA replication. So in this case now, if we have this as DNA strand A, T, G, C, G, then C, and we have this as a complementary strand G, C, G, C, A, T. So this is a strand of DNA. So if this strand replicates, it's going to be in such a way that this strand this strand produces another strand, this strand produces another strand, and once it produces this strand, then this strand also produces another strand. Or let me put it here. This one also produces another strand. It's in such a way that this now becomes the newly formed strand, then this other one becomes the newly formed strand. So this is strand 1 and this is strand 2. So what it means is that the offspring strand, the newly formed strand or the offspring strand, contains one parental strand. This is one parental strand, then one newly synthesized strand. So for every offspring strand you see is containing one parental strand and one newly synthesized strand. I'm just using these dots to indicate the newly synthesized strand. So this is the the method that is called a semi-conservative method. So, but we want to look at DNA replication. And first of all, I will list all the enzymes that are involved in DNA replication. The first enzyme is called topoisomerase. We have the helicase. We have DNA polymerase 1, 2, and 3. Even though the one that is mainly involved in DNA replication is DNA polymerase 3, I wouldn't want to go into others. Then the next one is RNA polymerase, which is called primase. RNA polymerase is called primase. Then we'll have the next one, which is called the ligase or the ligase. So these are the enzymes that are involved in DNA replication. So the first one is topoisomerase. Let's talk about topoisomerase. This topoisomerase helps in uncoiling of the DNA. You know, DNA is so long that it's usually coiled around the chromosome. If it's not coiled that way, that means that if you stretch the DNA, the cell cannot even contain it because it's actually long. So, but the DNA is usually coiled, and when DNA replication wants to occur, there's usually uncoiling of the DNA, uncoiling of the DNA. Just like you have a rope, and you want to cut the rope. The first thing you do is to, if the rope is twisted, against itself or coiled, then the first thing you do is to stretch it before you now use your scissors to begin to cut it. So that is what happens. That is what happens. And this uncoiling of DNA is done with the first one, which is topoisomerase, uncoiling. Then the second one is helicase. 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 By the time the uncoiling is done, then the next thing that the next enzyme that goes into work is the helicase. And the helicase, what it does is to unwind the double helix structure. So as you know that DNA is bounded, I continue to draw this thing now. It's wasting our time. You know that DNA is bounded, then GC. So look at this. So there's a bond that exists between these strands. So the enzyme that helps to break this bond, the complementary bond that breaks this bond is now the helicase. That's the function of the helicase. To break the bond, the hydrogen bond that binds the bases together. That's the function of the helicase. Then, apart from the helicase, when the helicase is done separating the strand, because what is joining the strand together is the hydrogen bond that binds the bases. So, if it's separated in such a way that we now have this, then the main replication can occur. And the main replication occurs through the help of the enzyme called DNA polymerase, especially DNA polymerase 3. And what the enzyme does is that once it binds on the strand, it's not released on that strand until it is done replicating the complementary strand. So once it binds on the parental strand, then what it does is that it's there's is a kind of production of nucleotides that will complement the nucleotide that is already found in the parental strand. So the DNA polymerase continues to add a nucleotide in at opposite direction of the parental strand. If you watch the YouTube video of how this thing happens, once the enzyme is here, as it's going, it's synthesizing. As the enzyme is going, is as the enzyme is going, that's the DNA polymerase is synthesizing the complementary strand. As it is going, it's synthesizing the complementary strand. And it's not released until it synthesizes the last complementary strand, the last base that is found in the complementary strand. And like I said, this is a semi-conservative manner. So that is the function of the DNA polymerase. But then it's important I let you know that we have how many minutes now? 43 minutes. Wow. So it's important I let you know that. Let me see if we can finish this within before 1 hour. It's important I let you know that there are two different, when it comes to DNA replication, there are two different kinds of strand. One is called the leading strand. The other one is called the lagging strand. The leading and the lagging strand. Leading and lagging strand. Leading and lagging. So DNA replication is continuous and easy on the leading strand. And I'll tell you the reason. The reason is because on the leading strand, there is a presence of the 3OH terminal. Don't forget I said that DNA is made up of 3 primer ends. 3 primer ends. If you have 5 primer end here, then here will be 3 primer end. Then if you have 3 primer end here, then here will be 5 primer end. So if you look at the two strands of DNA, one must be the leading strand and one must be the lagging strand. Now for the one that is called the leading strand, it's called the leading strand because it contains the normal raw material that is required for effective DNA replication. And that raw material is the presence of these three primer OH terminal. Three primer OH terminal. So if we have five primer here, then here automatically contains the three primer OH terminal. So replication using the DNA polymerase becomes easy and continuous on the leading strand. So this one is called the leading strand. The leading strand. But then replication is discontinuous on the lagging strand. Replication is discontinuous on the lagging strand. It is not direct. It does not occur direct. The reason because this strand does not contain the raw material required for DNA polymerase to work. DNA polymerase requires the three primer OH terminal. If you look at this strand now, because if here is three primer, here is five primer. If here is now three primer, here can be three primer again. It is now five primer. And this five primer does not contain the normal raw material that DNA polymerase requires to work. And since it does not contain it, the replication on the lagging strand, this one is called the lagging strand. The replication on the lagging strand is not continuous. Is not continuous. And is now the replication now occurs in form of fragments. This fragment is what they call the Okazaki fragment. Okazaki fragment. The replication now occurs in form of what we call the Okazaki fragment. The Okazaki fragment. So instead of going direct this way, the replication occurs this way. Because we are looking for the three primer OH terminal, and that three primer OH terminal is found at this edge. That means that the DNA polymerase will now begin to add. Sorry, not the DNA polymerase. The the RNA polymerase, which is primase, provides the terminal. Provides the terminal which the DNA polymerase will now use in the synthesis of this ligand phase. A minute. Let me. So, like I explained, the primase or the RNA polymerase will now provide the terminal, the three primer terminal. The enzyme helps in providing the terminal that the DNA polymerase will not work with. So once the terminal is provided by the primase or the RNA polymerase, the DNA polymerase will now begin to synthesize the 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 lagging strand. But it's now in the opposite direction. It's now in the opposite direction compared to the leading strand. And the replication is in segments. In segments. The replication is usually in segments. So this one we are done synthesizing this. So this occurs in segments, and in these segments I said are called Okazaki segments. Okazaki segments. But if this is how it is in the lagging strand, that means work done is still not complete. What happens to this species? Because a full strand is not yet formed, and that is when the DNA ligase comes. That is when the DNA ligase come. The ligase. So what the ligase now do is to fill up these spaces between the segments. And once the segments are filled up to form a continuous strand, then that brings uh, the process of DNA replication to an end. So this is how DNA is replicated. Okay. Then I didn't mention there is a little. Let me say something there. So I didn't mention DNA polymerase one. So the function of DNA polymerase one is to fill up this gap. No, I mentioned that we have gap because the function it was together with the ligase. It was together with the ligase. So what DNA polymerase one does is to fill up these spaces. Once the spaces are now filled up, then the ligase now connects the segments together. So DNA polymerase one and the ligase help in making sure that we have a continuous strand after the formation of Okazaki fragments. All right, I think this is where we'll stop because this video is becoming long. This is 51 minutes. This is where we'll stop. Uh, then before we go, okay, in the next class we'll go into gene concept. But just listen as I read out this. The process described above is typical of E. coli. So what we describe now is the DNA replication in E. coli, the bacteria called E. coli. All the enzymatic processes are generally the same in prokaryotes and eukaryotes. However, However, the process of DNA replication in two classes differ in the following aspects. Prokaryotes, I mean, looking at the difference or the differences between DNA replication in prokaryotes and eukaryotes. Prokaryotic chromosomes are circular, and there is a single initiation point of replication, whereas eukaryotic chromosomes are linear with multiple points of initiation. That is one difference. The second one, eukaryotic DNA, unlike that of